Hey guys, welcome to DNA, the molecular basis of heredity. This is going to be a really exciting unit. A few years ago, my mother, my sister, and I were interviewed for the Charlotte Observer because we were all teachers and we all came from one family. And my sister told a story of how one student was complaining that, "Why do we have to learn this, Miss Beaker? This is boring." And one girl raised her hand and said, "I don't know how you feel, but I look in the mirror every morning and I say, 'Thank you, DNA.'" So this is super important, DNA, and you can look in the mirror every morning and thank your DNA for it. All right, let's go ahead and get started. So why is DNA important other than just looking at yourself in the mirror? DNA, it stands for deoxyribonucleic acid. It's one of the two nucleic acids found in the body. And it is the blueprint for life. It codes for the proteins which determine traits and make up everything. This includes what determines your eye color, and those proteins are called pigments. It's more than just one gene, but it codes everything from your eye color to your hair color to how you look overall. So DNA is really important. All right. DNA is packaged into chromosomes. We've been talking about this for a little bit. A chromosome is a continuous strand of DNA. In humans, we have 46 continuous strands of DNA, 23 pairs, and they're tightly coiled around proteins. Those are called histones, and those protect them and allow for us to determine expression, all those different things. And we remember that chromosomes are found in the nucleus of eukaryotic cells. Prokaryotic cells have a single chromosome called a plasmid. It's circular and it's free floating in the cytoplasm. All right, so how did they discover DNA structure? So really we should give most of our credit to a couple different people. But so many scientists had to work really hard to determine the source of heredity. So heredity is the passing of traits from parent to offspring. And to determine that, a lot of work. So they understood heredity, but they didn't understand where did it come from. So first scientists determined that chromosomes controlled heredity, and that those chromosomes were made of what they call DNA and proteins. All right. Then over time, DNA was determined that it was the chemical that controlled the characteristics, not the proteins in the chromosomes. And then there was a race to determine the structure of DNA. So who did it? Well, most of the credit can go to Rosalind Franklin. She was the first to take a picture of DNA. She used a technique called X-ray crystallography, and this gave an idea of the shape. Rosalind Franklin is absolutely amazing. However, she was working with X-rays all the time, and she passed away not long after her work from cancer that she got from being exposed to X-rays so much. She never got the Nobel Prize for her work, because Nobel Prize can't be given to you after you are dead. So the people who got the Nobel Prize were Watson and Crick. They finalized the model and built a big 3D model. They're two Brits, okay, and they used pictures taken by Rosalind Franklin and work from other scientists. So they actually didn't do a whole lot of work themselves. They just put together the work of other people, and they came up with the double helix structure in the 50s. Okay. All right, let's look at what the structure looks like. All right, so DNA is a double helix. All right, it's a twisted ladder. All right, the building blocks are called nucleotides, but the nucleotides are made up of 3 different parts. First, we have a sugar, and the sugar in DNA is deoxyribose, OSE. Remember when we were talking about sugars, glucose, fructose, deoxyribose is a sugar, okay? Then we have what's called a phosphate group. You'll definitely learn more about that when you take chemistry. And then you have one of 4 nitrogen bases, all right? And they're called adenine, thymine, guanine, and cytosine, and we abbreviate them using their first letter, A, T, C, and G. Then let's talk some more about what's going on between A, T, C, and All right, so the two strands are held together by hydrogen bonds, okay? A and T hydrogen bond together. They have two connections between them. G and C bond together, and they have three hydrogen bonds, or three connections between each other. And those hydrogen bonds hold together the DNA, all right? And again, as I just said, A and T bond together with two attractions, hydrogen bonds, and C and G bond together with three connections. And this pattern is called complementary base pairing. Complementary base pairing. All right, so determining the complementary base sequence. All right, so this is one thing you're going to have to be able to do for the EOC. When you're given a sequence, you have to be able to come up with the opposite pair. But you have to remember that T bonds with A bonds with T, G bonds with C, C bonds with G. Okay, hopefully that shouldn't be too hard. So here we have a sequence. Let's go through and see if we can figure out the complementary base. All right, so T bonds with A bonds with T, G bonds with C, G bonds with C, C bonds with G, T with A, G with C, A with T, A with T, C with G. When you go to do this on the EOC or any practice work, you should simply write below the matching pair, and then you can match it to a choice on the assessment. All right. Now, this sequence only has 10 base pairs. 10. It's only 10 across and 10 base pairs. All right. The human has approximately 3 billion base pairs in your DNA.